Okay, welcome. Welcome, everyone. As you notice, it's kind of like the Methodist Church. You have to come early if you want to sit on the back row here. So you have to come up to the front now if you come late. We are going to be doing this streaming video again, so I'll ask you to mind your manners. I won't stand over there in front. I'll stand here so they can get it all. I'm very pleased to introduce to you our speaker for today, Tom Bianchi, a good friend and colleague from up at Texas A&M. Tom is a professor there in the Department of Oceanography. He holds the James R. Watley Chair of Geosciences that he was appointed to this year. His particular or special fields of study are marine biogeochemistry, organic geochemistry, and climate change science. I could give you a lot more of that, of his interest areas, but he's going to touch on those kind of things. He's really had a really productive and active career in publishing over 125 peer-reviewed journal articles, lots and lots of grants from NSF and other major funders. He's done 4 or 5 books and has a couple of more in line. I'll read you just a couple of the titles of those so you can see what they're about. Biogeochemistry of the Gulf of Mexico Estuaries, that was with John Wiley and Sons. Biogeochemistry of Estuaries, that was in Oxford University Press. Hypoxia of the Northern Gulf of Mexico, that you'll learn more about today. Another chemical biomarkers in aquatic systems. So just really a lot of biogeochemical kinds of things and a real focus on estuaries. He's really been involved also not just in the U.S., but in other estuaries of the world, from kind of Europe to Asia, and most recently really involved heavily in China. Some really neat activities over there. He goes over there quite a bit. He's been nominated as a AAAS fellow this year, still to see whether he gets that stamp or not, but it's a really high mark just to be nominated for that. Tom is originally from the Northeast. He got his master's degree under Jeff Leventon, the famous marine ecologist up there. Tom says that's where he filled one side of his brain. And then he went to the University of Maryland and got all the chemistry side of the other side of his brain. Then he went to Tulane University, had a very highly productive career there until Hurricane Katrina. And most of you know what happened in New Orleans at that time, at Tulane and other places there. He got flooded out, literally. He actually says he got flushed out of Tulane and then went to Texas A&M. Tom, we're really pleased to have you here.
Thank you. Good afternoon. Thanks, Wes, and thanks, Luis, for inviting me. It's long overdue. I should have been down to visit the Institute a lot sooner. I'm very jealous of your view and fantastic building space that you have here. It's absolutely wonderful. I'll be very depressed when I go back into my office. Having looked at Wes's office today, I was very jealous. But so I guess most of you have heard about the hypoxia issue, and we thought this would be a good topic for a lot of the students working in the Gulf. And so what I'll do is first talk a little bit about the general Mississippi-Atchafalaya system, give you a little bit of a background on that, talk about the hypoxia problem, how that came about, talk a little bit about the differences between these characteristics of river-dominated margins. This is an important thing I need to bring you up on because they're very different than the typical estuarine systems that we all think of, and this has made dealing with hypoxia a different issue that I keep trying to make a point about. And then just some of the management issues, which I'll talk about at the end. So we'll see what's at stake here and some of the players involved. These are just a whole bunch of different people that I've worked with. A number of these guys are people that I worked with at Tulane, and they were also victims in Katrina and have now moved on to different universities and so on, and then some of my students and postdocs and stuff over the years. So if if you haven't seen this word before, it's something you should be aware of, and this is what we ecologists or ecosystem folks now look at as the era that we're currently in, that is the Anthropocene. And what this simply means is that with any particular system that you go to, it's very, very difficult to find a system that's pristine or operating based on natural processes. And the Gulf Coast is particularly anthropogenically disturbed. It's really, really a tough place to find anything along this particular margin back here in the delta that has not been altered in some way. So when you think of the general characteristics of an estuary or a river or a shelf, it's very hard to go in most places around the world unless you go to some remote places and not find yourself in the Anthropocene. So a little background on river-dominated margins. These are the large margins of the world that deliver most of the salt to the ocean from the minerals that run across the continents. It's about basically the 20 largest rivers in the world deliver the majority of material that we see in terms of sediments and dissolved materials to the global ocean. So big river systems like the Ganges, Brahmaputra, the Amazon, the Mississippi, the Yangtze, and so on are big rivers that have huge impacts and are very dynamic systems that we're becoming more and more interested in because they give information about land use change and they potentially record information about what's happened on the continent in these high burial depositional environments. So they're very, very different than the traditional semi-enclosed estuarine systems. You can see some of the big systems here in terms of sediment discharge, water discharge, and where the Mississippi is on the list, which is the one that we'll be talking about. As Wes said, we've been doing a lot of comparisons between the Mississippi and the Yangtze, or the Chinese name of that is the Chanjiang, and so there's some really interesting comparisons to be made. That's a different talk, but the Mississippi, just again to give you a little bit of a background, is made up of these big basins. It drains about 40% of the United States. You've got the Missouri, the upper Mississippi, and the Ohio. All of these come together in what we call the upper confluence, where the rivers come together. And pretty much from that confluence all the way down to the Gulf of Mexico, it's leveed. And so that really makes this river already a very, very weird system. That means it can't flood. And rivers are supposed to flood. But because of all the problems we had, particularly with the flood of 1927, that changed the way people viewed the Mississippi and living on the Mississippi, not just New Orleans, but even in the upper basin. And so people wanted more protection and so on. So that's made the river very, very different, to have this essentially like a flume moving from the confluence straight down. And it's also changed with a lot of damming. There's lots of dams in the rivers, which is happening in China at a phenomenal rate. So if you look at the Mississippi complex, if you look at basically the bottom of Louisiana, that was all formed by the river essentially moving naturally. So rivers eventually go through what we call an avulsion, which is a switching. They want to switch paths over time. Typically happens between 500 and 1,000 years. And as they move, if you imagine a hose with a lot of mud coming out of it, wherever you hold that hose, you deposit sediment. So there's going to be sediment deposited there, and so on. So these are essentially, the entire southern half of Louisiana is all mud. When my son wanted to collect rocks when he was young, I said, you picked a bad place. There are no rocks here. It's all mud. And he didn't understand that. He said, what do you mean, Dad? There have to be rocks here. We can't be living on mud. And I said, yes, we are living on mud. And why would they build a city on mud? And I said, well, that's a good question, too. And they should have never built a city on mud. But essentially, you don't start getting into Pleistocene sediments until you get north of the lake. So this entire section, these are all the different old lobes that can be dated thousands of years back. So this whole area is about 7,000 years old. which is a blink of an eye in geology. Okay, you don't find most cities that large, situated on sediments that young. Okay, so this area is still sinking because essentially the reason it's sinking is because it's mud and it's still condensing, or what we call dewatering. So it's just like if you leave a container filled with mud for 50 years, you come back where you mark that mud line, it's going to be down lower and lower as that water, as that sediment condenses. So essentially this whole area is subsiding, which is making relative sea level rise a big problem here. So that's a little bit of a background on the large rivers and this particular one. Now, what about the story that leads up to hypoxia? Well, the first thing we see is that because of the construction of dams, you see the average suspended load from 1950, this is to 2000, the amount of particles in the water, okay, has started to drop. And the reason for this, you can see these numbers in terms of milligrams per liter. This is all because of the dam construction. So when you build a dam, you not only hold water back, but you allow the particles behind that dam to settle, which allows the water then to move on and carry nutrients still with it, but with much less particles. But it's been estimated that with all the dams in the world, we've altered, or we're holding up about 600, by 600%, we've altered or restricted the water flow from land to the ocean. By 600%, we've made a 600% reduction in the natural water flow from land to the ocean through dams around the world. Now, the other thing, as I mentioned, that's part of this story, so we have fewer sediments, but we have more nitrogen going up, 55, 2005. And again, you can see some of these numbers. And this is primarily because of the utilization of chemical fertilizers, particularly nitrogen, particularly nitrate, and the same thing's happening in the Jonge. In fact, the nitrate footprint, or the nitrogen footprint that people are now seeing, Bob Howarth and some other people, off the Yangtze and Yellow River is unprecedented. They thought the numbers were wrong. When you look at red dots as the size of the nitrogen footprint, there's a big blob covering most of the eastern part of China, and they thought that the numbers were wrong. So it's unprecedented, the amount of nitrogen coming into that system. So they're repeating what we already did. Even though they're an older nation, they're just sort of entering into this whole new industrialization, and they're kind of going through what we already did, so it's kind of interesting. But we have nitrogen coming down, and then, of course, if you look at discharge in the river, this is from the Ohio, the upper Missouri, upper Mississippi, and the Missouri. All I want to show you here, this is discharge recorded at Baton Rouge. Okay, so this just shows you the variability, the monthly variability. This is a typical, what we call a hydrograph, where you go through high flow and low flow. And this is to show you that most of the connection with flow downriver is from the Ohio. So most of the water that we see coming out of the Mississippi in the lower river is coming from Ohio. Most of the particles are coming from the Missouri. Another thing that's interesting, this is the water discharge at Baton Rouge, and you can see that at low discharge you get high chlorophyll A. And the reason you get high chlorophyll A is because the water is clearer at low discharge, and there are more diatoms and phytoplankton growing in the lower river than we ever realized. If you look at the typical numbers of, let's say, the Amazon in terms of chlorophyll and micromolar, you can see 0.17 to 2.38, and you can see here it goes 0.8 to 23.6. And we took a look at this and we thought maybe the numbers were wrong, but then it turns out that, in fact, there is this huge phytoplankton influence. And again, look at these numbers here in micrograms per liter. Upper Mississippi, micrograms per liter. Missouri, micrograms per liter. Ohio, and you can see the numbers get up near for those of you that know anything about sort of bloom conditions, these numbers are what you would see in normal phytoplankton blooms on the coast. And this is in a muddy river where you're not supposed to really have much phytoplankton growth, or at least most people think that. So there is a lot of phytoplankton. Because of the damming, you're allowing these phytoplankton, particularly in these carved off areas or these dammed regions, to grow. And then I think most of the material that comes down the river is in a senescent phase. So you see a lot of diatoms moving out onto the shelf, and then through osmotic stress, most of them are senescent. I don't think they're actively growing. You get a lot of deposition of diatom detritus right off the shelf, the inner shelf. And you can see that what used to be considered to be the big muddy is the Missouri is becoming green in many parts now because of these dams. Okay, so that gives you a little background on the nutrients and the particulates, and now we can lead into the hypoxia issue. And everyone, I hope, knows the general idea is that you bring nutrients in, you produce more algae than the system should be receiving. More of that material gets to sink because it doesn't get taken up by secondary production. As it's sinking, you get a lot of bacterial consumption, you get bacterial in the sediments. And then typically when it's equal to or less than 2 milligrams per liter, you have what we call hypoxic conditions. When it's 0, you have anoxic conditions. And you can see that this has an impact on marine life. So hence the big concern about these kinds of systems around the globe. And this is a problem not just in the US, it's everywhere. There's Hans Perl, who's now working over in China in Lake Taipu, sampling his, believe it or not, that's actually lake water. So it can get pretty nasty. Like I said, the nitrogen over there is just really amazing. So Hans has been having a good time going back and forth there over the last few years. And then you can see a lot of the other systems in our neck of the woods that are also impacted. So no problem, no doubt that that's a serious problem. If you look at hypoxia in the Louisiana system, this is pretty much, it started to monitor this back in 1985. It wasn't really discovered by Rabelais. It was discovered by some other folks in the late 70s, some benthic people that were sort of sampling around, but then Nancy was the one who really started going out and sampling every year. And what they do is they go out and they sample the same stations, the same time, every year. And that's what you usually get a report on each year, whether it's the size of Connecticut or Massachusetts or whatever. They usually give some sort of analogy to give people an idea of what that looks like in terms of the size of a state or whatever. And one of the interesting things is that, when you start looking at these changes, one of the things that really jumps out is the 1993, we call this the big jump. All of a sudden, there's a big jump here. Yes, there's a couple of low flow years that were very low river flow, but for some reason, the data just all of a sudden appears much, much higher. And I'll talk a little bit about what Gene Turner and others have interpreted that to be. It has to do with a legacy effect on a paper that they published called the legacy effect. But this is what the hypoxia region looks like. And again, from all this data, you can see the hot spots. These are the areas that are most frequently, when you go out in any given year, greater than or equal to 75% of the time, that region is going to be hypoxic. Now, the problem that I really have, and you'll hear me say a lot of negative things today about the science that's been done here, is one of the biggest issues I have is that when people talk about the hypoxic zone, and I don't like calling it the dead zone at all, and I'll tell you why about that later, but let's just call it the hypoxic zone. Even that in itself, when I talk about the hypoxic zone, I talk about the near field, midfield, and far field regions of the hypoxic zone, because this is really the hot spot between the Atchafalaya and the Mississippi, because it gets nutrients from both. and it's a hot spot for deposition. But when you talk about over here, I mean, the mechanisms over here are very different. If you get into the nutrients here that are coming out in the Mississippi River plume, there's no doubt in my mind, I'll say it very clearly right now, there's no doubt in my mind that the hypoxia here is definitely from the Mississippi River nutrients. Okay, I said it. But now I'm going to say that once you get beyond here, the nutrients just strip out. So all of that goes into phytoplankton, the phytoplankton sink, and then they contribute to this hypoxia. The other thing I want to mention is everyone has this view of this horribly putrid, smelly, disgusting region. The hypoxia zone is typically about that high off the bottom. The rest of the water column is fine. So keep that in mind as well when you think about this region. Now, like I said, the mechanisms controlling this and how sporadic and how frequent. The other problem is going out once a year at the same time. We've gone out the week later and have very different numbers. So these are very dynamic systems. So that would be like looking out the window in May on a given day and saying every whatever day, May 5th, you do that every day, every May 5th each year. And you use that day to tell everyone what the weather's going to be like for the rest of the year. Okay? That's kind of what that's like. That's how bad it is. Because we go out and it's different. It's so dynamic that I'll show you some things that change within hours. So hypoxia events are things that have been well documented in estuarine systems. This is where the idea came from. I think in 1985, when we discovered it down here, Nancy and others, they said, this has got to be the same thing that's associated with Chesapeake Bay and all of that. It is semi-related. However, these systems are semi-enclosed systems that have much more restricted flow. And when you get into the region and you collect a mud sample from the middle of the Chesapeake Bay, as I did when I was a graduate student, it is the most horribly black-smelling, sulfitic, just nasty, nasty stuff that you can imagine, because they really, really get serious problems there where you can actually see it in the sediments. The other thing that was really evident from these systems when they first started going hypoxic is that you had a lot of dead things. So there were dead fish. I mean, it was really a significant thing. The seagrasses started to die because you had epiphytic algae blocking the growth of the seagrasses. That affected. So there were clearly linkages with the ecology of the system. We don't see that in this particular system. You can see that there have been shifts in the regime shifts with respect to some of the trophic dynamics of these systems. Very well documented, no problem. Okay, if you look at the effects of hypoxia on the benthic community, most people will go and they'll say that, gee, you know, the benthos in these systems are very small and they look like opportunistic or selected species. That must mean that the system is stressed because of the hypoxia. And you typically don't get, like any of you who've taken these courses, where you get these late-stage, deep-burrowing, healthy kinds of macrobenthic kinds of species out there. They're typically smaller, right in the hot spot of the hypoxic region. But even outside of that hot spot, it's the same kind of smaller species. And again, a lot of people, like Don Bosch and others, have attributed this to hypoxia. But if you go to most other places where you have high deposition in these big river systems, the deposition is so high, the sediment deposition is so high, that that's typically the kind of species you get anyway. So I don't think there's a very clear linkage with benthos and hypoxia problems there. Again, I think the communities that you see there are what you see off of the Amazon, off of the Yangtze, where it's just too much deposition for case-selected species to be happy. This is what the mud looks like, and you should be saying, this must be the wrong slide. But this is what the mud really looks like, and we've poked more holes in the bottom of that map that I've seen you than anyone else in the country in terms of the mud samples that we've collected over the years. Because, as I say, when they go out, do their survey, they're just doing water column surveys with oxygen. So the one thing that should be obvious to you if you're not a mud person is that this looks very oxidized. That is, it has an orange look to it, like rust. That means that it's iron in its oxidized state, the iron oxyhydroxides. What makes mud black is the iron sulfides. That's when it's in a reduced state. Now you might say, yeah, but that's the surface, and if you go below, right below, you're going to see a dirty, black, smelly mud. No, you're not. And I'm in the hypoxic zone. So that seemed really weird to me when we started going out there and doing this. What is going on? How could this really be viewed as such a negative effect on this system if these muds look so normal? Well, what about the fisheries? Well, except for Menhaden, and Menhaden, you can see that there's a slight decrease. All but Menhaden, basically the fisheries have gone up this whole time in the hypoxic region, the dead zone. So all of these systems have increased. Now, you might say, yeah, but what about the Menhaden? Well, that's primarily due to overfishing. So even the Menhaden doesn't fit that story. So this is very puzzling. How can we have a fairly normal benthic community, an increase in fisheries, and still be spending so much money on why we have this so-called dead zone? And this is from Jim Cowan, who has worked in this system and has sampled continuously out in the hypoxic and finds no regime shift either. So you might say, yeah, but it increased, but are the species changing? No. So we're not seeing that either. This is still considered to be, in the Gulf of Mexico, the Fertile Crescent. Okay? As I was saying earlier today, most of the bycatch from Louisiana is greater than most of the other states combined in terms of their actual catch. So it's hugely productive. So, to date, there's really no clear evidence. Now, some of you might be saying, well, I heard something about croaker having problems with gonadal development and things like that, and I think some of that work's have been done down in this region, and I'm not sure the details, but that's true. And one of the things you have to realize is that croaker reproduce at the mouth of the Mississippi, and the Mississippi is one of the most estrogen receptor contaminated systems in the country, and so I don't see how the linkage with hypoxia can be linked with gonadal development problems with the croaker when they're reproducing near the greatest source of estrogen receptor disturbance systems such as the Mississippi. So I think, in my opinion, that new work that's come out is very interesting, very well done, but I think the source of the problem is the river, not the hypoxia. So what about this river-dominated margin? What makes it so resilient to hypoxic conditions? Why is it not like Chesapeake and Baltic and Long Island Sound? Well, there may be other types of controls other than this nutrient-centric view has been overlooked. Anytime you bring up any other potential factor that can affect or control hypoxia, the people that have been selling the idea that it's all from the river get very upset. And that's where we've had our problems. So, missing data. Recent action plan by EPA has shown that there's very little work that's been done in the 20-plus years of NOAA funding, because most of the work, as I said, has been documenting the size or the monitoring of the hypoxic zone. So if you look at all the work that's been done in Chesapeake Bay, denitrification work, nitrogen fixation, Delaware Bay, all of these places, huge amounts of data on that. And you might think the same thing exists for the hypoxic zone. It has to exist. It doesn't. It's unbelievable what's lacking there in terms of the most basic things that are related to redox processes. Why is this? Well, one is that most of the focus has been on press releases. Most of it's been on getting a lot of the attention to this region, which is good in one way, but then eventually you have to move on and start doing the real mechanistic kind of work. And that just has not been getting done. We're finally starting to do some of that now, but it's amazing when you think that this has been going on since '85. And again, these are the kinds of things that, you know, it's usually more reporting the size and the state, which again is fine if you have other types of mechanistic things going on. So I think that's one of the problems. So there are 4 natural and anthropogenically derived characteristics of these systems that I think are making this system less vulnerable than other estuarine systems. One is the spatial and temporal variability of the freshwater inputs, the diversity and magnitude of organic matter sources, the rates and efficiency of organic matter cycling. We have mobile muds. What are mobile muds? They're things that you typically don't find in most other estuaries that you find in most large river margin systems. I'll tell you what they are in a minute. The other thing we have is we have transport mechanisms that are way beyond what you're going to get in Chesapeake Bay or Delaware Bay. We have the ability to dump a bunch of organic matter in the Mississippi Canyon. We have the ability to move things all over. That also makes the organic matter that's accumulating, typically in a more semi-enclosed estuarine system, a whole different story on the open shelf. First, the temporal and spatial variability. This is just showing you August 5th, August 13th. Essentially, this is some work from Hetland and DeMarco. And in one system, you have upwelling. The other system, you have downwelling 8 days later. And you can go back 5 days later and find it different. So in terms of the dynamics of circulation, very, very, very complex dynamic system. Not that you don't get that in estuaries, but this is really much more complicated by the large freshwater plume. If you look at the Texas coast, we have recently, this is a DeMarco et al. Paper, just came out this year. We have looked at oxygen isotopes, and one of the interesting things is that as you move further west, everyone has always attributed some of the hypoxia that you see in northern Texas to what's coming from the Mississippi. It's not to say that there's never an influence on that, but what this shows is that you can have hypoxia, and you can separate these different water masses using oxygen isotopes. And the Brazos was essentially the reason for one of the big hypoxic events that occurred a few years ago, and it was not related at all to the Atchafalaya, Mississippi outflow. So again, it shows you that the dynamics of these coastlines that are right next to each other, that many people feel it's just sort of one big hypoxic region. The diversity of sources, well, if you look at chlorophyll A in the sediments and you look at fucoxanthin in the sediments, this is a biomarker. This is a biomarker for phytoplankton in the sediments and diatoms in particular. No surprise that this is the hot spot. This is where you find a lot of the hypoxia. This is where a lot of the diatoms settle out. And no surprise, this is exactly where it is being fueled by the river. Once you get outside of this, the nitrate goes way down, primary production goes way down, and you cannot say any longer, once you get west of here, that it's nutrient-driven hypoxia if you're going to go to the Texas coast, the border where they go to map it every year, and attribute any hypoxia as being the same mechanism for that whole system. If you look at some work that we did, and by the way, this is what the plume looks like if you've ever been out. You can see it very sharp, the Mississippi River plume. So I had a postdoc work on this, Rebecca Green, and what she did was she took the area, the particle plume area, and this is the whole hypoxic region, and tried to look at if you could take all the productivity from the river nutrients that are producing the high activity of the plume and really contribute all of the drawdown of oxygen. And it turns out that basically it only contributes about 23% of the demand necessary to fuel the whole hypoxic region that gets mapped every year. Okay? So something else is fueling the hypoxia that you see, albeit less frequent, to the west of those hot spots. And no one wants to hear that. Everyone gets very upset when you say that, because while I understand that they want to make reductions in the river, you can't say that everything in the hypoxic region is being caused by nutrients in the river if it's not really being caused by that. As much as that may be a diversion from the work that they want to get done up in DC, you have to still acknowledge that this region here may be controlled by other things. Well, what about DOC? DOC is a really good way to bring oxygen down. You don't even have to make phytoplankton. Nutrients, you have to do two mechanisms. You got to bring the nutrients in, you have to make phytoplankton, and then you have to get the microbes to eat it. DOC, you can just bring it in and the microbes eat it directly. You don't even have to grow phytoplankton. Is that ever considered? No. Is DOC a big issue in the Gulf of Mexico? Well, DOC has, in the Gulf of Mexico estuaries, some of the highest DOC numbers you'll find in the country. They're very DOC-rich systems. What I'm showing you here is if you go from the marshes out to Terrebonne Bay, out to the shelf, you can see big DOC drops. And the other thing we've measured in this is lignin. Lignin is a tracer for terrestrial plants. So we know a lot of this DOC is actually coming from marsh plants leaching as they die and go through various cycles. So we know where a lot of this DOC is coming in, and I think that that's a big factor that's being ignored that may be fueling some of this periodic, sporadic hypoxia to the west. So one of the arguments has been, no way, because microbes don't like to eat terrestrial-derived material. They only like phytodetritus or phytoplankton-derived DOC. Do they like it better than terrestrial-derived DOC? Yes. Is it more digestible than terrestrial-derived DOC? Yes. Does that mean terrestrial-derived DOC is not consumable? No. We had a flood back in 2011 where they had to open up some of the systems they hadn't opened in 30 or 40 years in the Atchafalaya. Huge plume of water went out from the Atchafalaya to save New Orleans. They flooded all the Cajuns. They weren't very happy about. And what we show in this paper is essentially that the DOC that went out is terrestrially derived based on the biomarkers, based on stable isotopes, and that there was huge flux of CO2 to the atmosphere, which tells you that the DOC was consumed by bacteria, because we also, through RNA work, saw that there was a change of bacterial populations just within 2 weeks. It's amazing how quickly they change, just like they did with the oil spill. And they change to microbes that eat terrestrial-derived organic matter more than the ones that typically eat marine. So when you get flushing of marsh-derived material to the coast, can it be consumed by microbes to reduce oxygen? Yes. This has been shown. This was a perfect experiment for us to really prove that terrestrial-derived DOC is in the game. What's another different thing about these systems? Well, it's not just terrestrially derived material. We also sampled all in around here. This is in the sediments. And we did a mixing model, stable isotopes, to look at how much of the material in the sediments is coming from different sources throughout this region. And you look at this and you see this is basically in spring, this is fall, this is the region. And you can see that there's a fairly significant fraction, particularly in spring, almost up to 40% of the material that's coming from terrestrially derived material. Some of that can be from the river, which is actually coming from soils all the way up in the Midwest, and some of that can be from local marsh material. And that's not an easy thing to separate, but nevertheless, it's terrestrially derived and being ignored, okay? Totally being ignored. Well, if you look at historical changes in hypoxia, and you can do this using this PEB index, which is using forams that are more adaptable to low oxygen or less adaptable to oxygen. And you can go down core and you can look over time and see how the forams have changed with changing oxygen conditions. So if you look at this, you'll see that from these different cores, basically this means that oxygen is decreasing when you have a greater abundance. And what this means is that since 1950, you're finding a greater abundance of forams that look like they're living in a low oxygen environment. Perfectly normal. This was taking right in and around the hotspot. So that fits pretty well with the increasing nutrients that have occurred and with the whole idea of plume-derived nutrients controlling the near field part of the hypoxic zone, which, by the way, is a very small part of it. There are the nutrients again. I'll just show you. This is from a different data set, so that fits nicely with that trend of increasing. And the other thing, though, that's happening is not just increasing nutrients, is that we've had huge amounts of wetland loss, totally ignored in the picture. I don't know why people have ignored this, but there's huge amounts of material being transferred from the coast because of this mostly highly erosive system in the form of DOC and POC, sediment, organic carbon, and it needs to be factored in when you look at oxygen consumption. If you look at how far hypoxic events go back, we actually see that they go back to 11,000 years before present, based on the foram data. So you say, gee, that doesn't seem very nice. How can that be? Why would this region be hypoxic 1,000 years ago? I thought the nutrients started to increase only in 1950. Well, one thing is to consider is that this is a depositional system. This is a high depositional system. I told you it formed the entire region here. So you can imagine what's underwater here. These systems, probably 1,000 years ago, were undergoing greater riverine inputs and probably lots of organic matter flushing down, lots of stratification, and you could have hypoxia in the absence of nutrients. That's a really bad thing to say in front of some people. So you can have hypoxia in the absence of nutrients, and that is definitely something that is clear here. Well, we talked about this material maybe being too refractory, and I proved to you that the terrestrial DOC is not. We showed that recently with the flood. What about the stuff that's in the sediments? Well, I talked to you a little bit before about mobile muds, and mobile muds are really these fluidized muds that accumulate. They're about 10 centimeters deep. That was in the box core, which is why you don't get black mud below them. They're constantly being resuspended. That's not what you get in the middle of Chesapeake Bay. You get a lot of deposition, very little mixing, and oxygen consumption. One of the things that Bob Aller and others have shown is that if you look at this global incineration, that is the amount of carbon that's consumed, mobile muds are some of the best muds in the world to digest organic matter. So this is a great system where you're not accumulating material, you're digesting it, and you're probably digesting terrestrial organic matter really easily. because the more it's mixed, the more oxygenated the reactions are, and we know that oxygenated reactions tend to be more efficient than anoxic reactions. So this is another thing that makes this system very different than the other types of systems we can compare it to. And I'm just going to show you quickly here. This is just mobile muds moving out across the seabed. And if you look at some excess lead-210, I know this is more of a marine biology audience, but you look at stable isotopes to show you the depth. When it's vertical, it just means it's well mixed, and we've used beryllium and lead-210. And you typically see the mobile mud zone about 10 to 15 centimeters deep. And that just sort of sloshes around, gets resuspended, moves out to the canyon, moves west, moves all around, basically like a chocolate pudding. One of the other interesting things that we see in this system is that the acid to aldehyde ratios, I'd say what in God's name is that? Those are decay ratios of lignin. So this higher that is, the more lignin you're seeing degraded. That means the more terrestrial organic matter is being degraded. We see a really interesting linkage with where we see hotspots in chlorophyll A. We see a lot of lignin decay. And I published a paper in PNAS last year that we just got some funding from NSF. They're very intrigued by this. We view this as like a priming process. Priming is where you take very labile organic matter and you mix it with refractory organic matter, and it breaks down faster. This comes from the literature in agriculture back in the 20s when they wanted to make hay more degradable, and you can mix alfalfa and things like that in with it. So you take the goodies and you make the ones that are difficult to break down for organisms more digestible. Very, very important in terms of carbon sequestration, CO2 cycles on a global scale. And the soil people, as I said, have led the way, but the aquatic people seem to be behind. So I think we're seeing priming here. And that's what this is, co-metabolism or priming, where you can see how far back it goes in the agricultural literature. So taking the terrestrial material, mixing it with labile, and you get higher turnover rates. So we now have funding to actually go and do a proof of concept in aquatic systems with some microbial folks. The other thing that's really weird when you go out in the hypoxic zone is that you don't see H2S. H2S is hydrogen sulfide. That's what smells like rotten eggs when you go near the marshes at low tide. Many of you know this. And it's also really weird because most of these anoxic systems in the estuaries are just absolutely loaded with it. So why do we not see that? Well, it comes down to chemistry. One of the other differences with this particular system, being a river-dominated system compared to a typical estuary, is that when you go to the big rivers, what are they draining? They're draining the continents. What's a really important mineral from the continents? Iron. There's a lot of iron coming out. When iron comes out and reacts with seawater, sulfate, right? That's one of the dominant ions in seawater. It's about 26 millimolar. You get sulfate reduction, and you can get the formation of iron sulfides. So it's kind of strange that even though I was saying before, we don't see the dark mud, there is sulfate reduction going on, but it's going on much deeper. And it's not really impacting what's happening at the sediment-water interface, because that's all oxidized by the mobile muds. So this is from work that John Morse did, and you can see that there's a great correlation with the amount of organic matter and the amount of pyrite. And I would say about 90% of all the pyrite formed in the Gulf of Mexico is formed off of the Mississippi River. So it's a huge sink for pulling sulfur out, and we think that, again, a lot of that gets trapped in the minerals. Okay. What about transport? How is that different that I mentioned before? Well, if you look again at spatial variability of surface deposition, this just shows you, based on beryllium-7 inventories, no surprise, right off the mouth of Southwest Pass, you get huge amounts of deposition of sediment. It varies seasonally, but this is the bullseye, and this is where the action is, where the real hypoxic zone should be restricted. When you draw a map of the hypoxic zone, this is really about how big it is. The other part to Texas is, again, not linked with the river. But what about organic matter that accumulates there? Does it stay there? Well, this is a very busy slide, but I'll just show, I'm not going to drag you through all of this. If you look at the material out in the canyon, this is the Mississippi Canyon. Okay, this is where, notice it's pointed this way, because the river used to feed into it this way in one of its older positions. But we went out into the canyon and said, do you see any material out here that looks like it came from there? Because maybe there's this like flushing mechanism. Maybe that's a great way to really keep the system clean, unlike what you can do in most estuaries. You have an open coastline, and maybe it just like exports a lot of this stuff, and it doesn't stick around and accumulate like it would in a lake or more stagnant estuarine system. Well, sure enough, when you look in the sediments here, first of all, you see beryllium, which tells you that there's sediment getting out there very quickly, because you don't see beryllium in deep sea sediments unless it's getting out there from the shelf, and I won't go into the details. You also look at biomarkers in the sediments, and you look at the primary production above these waters, which if any of you have ever been out there, this is real blue water, and there's no way that you can support the amount of phytoplankton that you see in the sediments here from the water column. So it's being advected out. How does it get there, and why does it go so far? Mobile muds. Another thing about mobile muds is that not only do they efficiently incinerate organic matter, but they move around, particularly when storms and come through, and you can essentially move that material out. Sorry. So the management plan, this is the last section. Keep in mind that if you don't do things right, and you just sort of report on the same thing year after year without doing the mechanisms, and looking at the mechanism, there's going to be big problems when you get to management issues in terms of telling the farmers to essentially go out of business in terms of controlling nitrates and how much to control and how much to reduce from the river. When you think of the work, probably the best work that's been done in terms of Chesapeake Bay, you can look at, that has been worked on for years, the most well-studied estuarine system probably in the world. That's because the senators park their boats very close to it, and they like funding that particular estuary. But it's a very well-studied estuary. It's had problems. But the Save the Bay effort, which is on all the bumper stickers and all of that, has failed miserably. You can see billions of dollars have been spent, Save the Bay, and 30 years ago, and they still get a grade D. And they measured everything. Sulfate reduction rates, nitrate reduction, you name it. Every kind of biogeochemical process has been described to the maximum there. Nitrate deposition from the atmosphere, all of these things which have not been measured in this region, the Gulf of Mexico. So the thing is that you have to be very careful about what you promise and what you're telling people about in terms of the danger of it. And again, they had a real good reason to go after it. And they did improve the water quality. Some of the seagrasses came back. But in terms of really understanding how to manage how much nutrients go in and how much nutrients are coming from all these different sources around the bay and so on, and all of that's been really, really detailed budgets. They still have a problem managing it. So that's the best case scenario. So I was asked to get involved in the 2007 hypoxia assessment team. And in 2001, they met, the team before the one I was involved with, and they used a 5-year running average of 5,000 kilometers to be reached by 2015. So in other words, when you see 21,000 square kilometers that Nancy might report for a hypoxic region, they're saying we need to get this down to 5,000 square kilometers by 2015. That's what they said, 2001. Stop doing that. And that would require a 30% reduction in what's coming out of the river to do that. That's what they believe. So when I went to the meeting in 2007, the people at NOAA, PhD people, said, our main objective here is to figure out how to get this system down to 5,000 square kilometers. And I raised my hand and I said, where did that number come from, and what does that mean in terms of what we get in return? Does that mean an increase in some fishery? Does that mean the shrimp come back, or whatever? So no one knew the answer to that, which was in fact, they took a coffee break very quickly. They were really stunned. I wasn't trying to be nasty. I just really wanted to know if they knew something that I didn't know. I said, where did that come from? Well, this number really kind of came out of the air. These guys in 2001 said 5,000 is pretty good. It's kind of a running average, sort of. And then let's just try to get down to that. But that doesn't give you anything. I mean, what does it give you? As I said, there's no evidence of any real negative effects. So you can't even argue that there's dead fish all over, and getting back to 5,000 will give you those fish back. So I said, why are we doing this? I mean, I said, really, I don't understand. What is it? Why are we doing this? And they didn't like that. So they also agreed that we have to go for the 5,000. They told me to shut up. And then they said, but we actually need a 45% reduction, not a 30%. And I said, you know, this is really kind of scary. A 45% reduction would really change dramatically what the people are doing in the upper drainage basin. Now, I am not in favor of more nutrients, and nor am I in favor of not making some reduction, because I truly believe that there are too many nitrates in the river. I'll state that right now. But I think you can make the argument for reducing nitrogen in the river more for water quality issues upriver than you can downriver. There are lots of problems with drinking water when the nitrates are that high. So I think you can make the drinking water argument better than you can if you try to link it to hypoxia. So I do believe you need to reduce nutrients in the river, but you can't play games like this and guess numbers and then come up with huge impacts on the farming industry. Recent estimates have shown that a 30% reduction in riverine nitrate flux would only result, this is from my postdoc, a 19% average reduction in plume primary production and 16% sedimentation rate. So that's not going to do very much if you believe that that's really the problem. So saving wetlands or reducing the hypoxic zone, truth or consequences. Again, I think the people in Louisiana are being misled. They have a much, much bigger problem. Their state is washing into the Gulf of Mexico versus a hypoxic region that's very productive, but someone tells them it's very bad, but we don't know why it's bad, but we need to reduce it because it must be bad, because hypoxia is a bad thing, which it is in most places, but you can't just go on the assumption that hypoxia is the same in every region, has the same effect. So truth or consequences, which one do we work on? Because what's happening is that this is getting ignored, and there's been more focus on the hypoxics issue. River diversions, as many of you have heard, there are a few of them. What they want to do is open up parts of the river to save and start feeding this area with mud, because the river is supposed to overflow. That's essentially how you grow a delta. It floods, it brings river water, the mud settles. So maybe we can open up some of these areas and allow it to start feeding mud, but you really need also sand, and you don't get that from opening up these gates. So that still has its problems, but am I in favor of this? Yes, big time. Some people actually think that these river diversions may exacerbate eutrophication in the marsh. And I really don't understand that either, because there's no marsh there anymore. It's essentially an open water system. So I don't really understand why they're worried about eutrophying the marsh when it's open water and you need mud and you need to plant more marsh to get the marshes back. So again, that's been another thing, nutrient-centric fear that's impeding. What's in the area? Well, let's come back to this big jump. The big jump, as we refer to it. Gene Turner published a paper. It was Paper of the Year in ES&T 2008. It was called The Legacy Effect. And the legacy effect, and I really don't understand why this was viewed as such a novel thing, because the legacy effect, as the way he describes it, is that if you keep polluting an area, because it keeps accumulating that material, you need less each year because it already has some from the year before. That makes sense. And this is really the basics. I mean, there's nothing new about this. This is essentially the basics of limnology, which was described back in the 1800s in terms of how lakes fill in because they become eutrophied and they eventually choke up and fill in. So that, there's no need for a new name. It's been around for a long time in limnology, that systems fill in from previous organic matter accumulation, and if it doesn't get flushed away, you fill in, and eventually lakes become eutrophied. In fact, eutrophication is a natural process in lakes before humans were ever around. They just eventually choke up. So in limnology, eutrophication actually means something very different. It's part of a natural process in terms of the evolution of lakes. So I wasn't too impressed with the legacy effect. And the other thing is that he says that this is why there's a jump at this time, and except for these low river flow areas, this is why we get this. Well, this has gotten really even more confusing. DeMarco and Don Scavia have recently reported that Nancy's been calculating the area in the wrong way. GID protocol? No, I didn't mean GR, I meant GIS. This is something I'm not, thank heavens I'm not involved in this, but I'm kind of just staying out, hearing things from the wings. But they cannot reproduce the areas that have been calculated all these years. And well, some of this I can't share with you because the NOAA folks are very, very upset about this because they've spent a lot of money on this and now they're thinking, what is it that we really have? Well, one thing that we do think is that maybe they started calculating area differently, but it's no legacy effect because we had really low flow this year. And if the legacy effect was so bad, this is 2003, remember we're in 2012, the legacy effect was so bad from the accumulated material in the bottom sediments, you would still have hypoxia even if you turned off the river, according to what Gene was saying. And you don't see much hypoxia out there this summer at all. So that legacy effect doesn't seem to be working. And as I said, I think that there's a problem in that. The reason why I also don't think the legacy effect works is because of what I said to you before. The mobile muds are an incinerator, unlike what you get in the bottom of a lake, and they burn up most of the organic matter. And even if they don't burn up most of the organic matter, a lot of it gets transported, flushed into the Mississippi Canyon, which also affects your whole concept of a legacy effect. Boy, do they hate me. More recent work has shown that from 1993, about 30% of the variance in the area can be explained by nitrogen loading, 54% by wind. These west winds result in a smaller hypoxic region, and the river flow is not significantly correlated with area when combined with the west wind duration. Over 85% of the variability can be explained by this wind effect. And this is work out of DeMarco's lab. She is now a postdoc up at VIMS and worked in Steve's lab modeling this. So that's something that I never even thought about. I'm strictly a biogeochemical person, but when you start getting into the physics of this system, it really gets crazy and it really gets complicated. And to say it's all linked with the river nutrients is really not very realistic. So as a realist, this is Sir Karl Popper. If you haven't read Popper's book on objective knowledge, it was something Jeff Levinson made me read. A philosophy book, a really confusing book to read, but he talks about falsifiability, and you look at what he says here in terms of search for the truth, highly informative theories look upon criticism as the main in promoting growth of our knowledge about the world of facts. And unfortunately, anything new that's been brought to the table about this system in terms of physical or other mechanisms that are not nutrient related to the river have not been viewed this way. They've been viewed in a really, really negative way, and it's been really unfortunate because it's really interfering with the work that's getting done there, and so on. General conclusions: are nutrient and phosphorus reductions needed in the Mississippi River? Yes, but for a much smaller region, and throw the 5,000 out, no one knows what that number means. Basically, I think we should make reductions and then start looking at the hot spot, the red spot, and see if we start seeing differences in the frequency of hypoxia with some reductions. But start off low. Has it worsened over the past 2 decades? I don't think we know. I think we need to first check and see what the areas look like. So, like I said, that's a big no-no right now and has a lot of people taking ulcer tablets. Are there unequivocal results showing the negative effect on regime shifts? No way. Not in my opinion. No way. No way. I think this system is much more buffered. Do I think we should ignore it? No. As I said, I think we should make some reductions and not refer to that whole big system because I think that's meaningless from the river mouth to the Texas coast. Absolutely meaningless in terms of what those cruises have shown. Are nutrient management issues in RIOMars different from traditional semi-enclosed estuaries? I don't know if I convinced you, but you can see I think they are. I think they're very, very different systems, and I think they have ameliorating effects. Much to our benefit. Are all shelf environments less vulnerable to hypoxia? No. It might not be the same. There's certainly low oxygen regions that are out in open shelf regions, these suboxic zones, no doubt. But mobile mud areas where you can dump things in canyons and other things like that, yeah, I think that they do have ameliorating effects. Has the emphasis on simple models, I didn't really get into this too much, I left this in here, but one of the other criticisms is that DeMarco and Hetland and some other people in our department at A&M have favored the more complicated models to include more of the physical forcing and winds and things like that. And we've been criticized because we've been criticized by the other team saying that, we just want a basic thing like river flow to correlate with that. Well, if it doesn't work, you can't do it that way. And sometimes simple models just don't work, even though that's what the managers want. If they don't work, then that is just a problem with, as I say, Occam's razor, reducing things to too much simplicity, and they just don't work. And it's unfortunate that no one wants to deal with what we call the big, scary models. I don't do them, but those are the ones that are needed for this kind of system. And finally, have political issues obfuscated our objective knowledge of research? I think you know what I would say to that. I think it's really been unfortunate that the Gulf is really viewed now, and these groups are viewed by other people all around the country as kids sort of fighting in a sandbox. It's just not productive. And so, we're trying to get together, and we're trying to work more. and try to coordinate things more. And I think we're headed in the right direction, but this area issue is going to not be a good thing until we get that resolved. So I'll stop there.
Tell Tom is passionate about these things, and he went on a little bit long, and so we've got time for maybe a couple of questions, but I've got to rush him to the airport pretty quickly.
Luis.
So what if you let the river flow naturally?
Hmm?
What if you let the river flow naturally?
You mean just leave it alone?
Don't give the flow rate long as possible.
Well, I mean, the problem with that is
I know people get into
no, the problem with that is that I think we have to start interfering with that because of the diversions. I think we need to start growing the coastline, or New Orleans is going to be on the coast. I mean, it is headed that way already, the rapid loss of land. So that's much more important. Forgetting about river diversions and making more terra firma, if you just leave it alone, I think it'll just continue to buffer itself. I don't think there's a legacy effect going on. I think the system will cleanse itself over time. I think there are mechanisms that will just leave it alone, and essentially we're not seeing the effects on the fisheries. The fisheries are still doing fine. Again, I have my issues with the croaker, and some other papers, even some other people have argued that, well, this hypoxic zone causes fish to move around it, or zooplankton to move, and energetically that's not favorable. And all. It's this high off the bottom of the water, and it's in the 20-meter isobath, so really you're not talking about most of the water column. So I think if you left it alone, and I wouldn't really want to say that in front of politicians, but I think it probably would just keep doing what it's doing. Remember, we don't know why that jump happened, that 93. We still don't know. We've asked if they calculated things differently, but that's going to eventually come out now that this area issue has become a problem. I think we're going to eventually find out what happened with the 93 jump, because that just doesn't make sense. Yeah.
You're not a fisheries scientist, but you served on a number of these panels. Has there been any discussion about the potential effects of reduced nutrient loads on fisheries? And Scott Dixon had been proposing talking about oligotrophic fish.
Oh.
And not only discussing the fertilization effects of nutrients
Mm.
on fisheries, but what happens when you start to pull back on them.
Yeah. No, that's a really interesting question, because I said this is considered to be the Fertile Crescent. I mean, this is the big granddaddy of the Gulf. What we throw away is scraps compared to what all you guys get in Texas and Alabama and everywhere else. So it's unbelievable how productive, and it's because of the productivity of that plume region. Now, do you need that much nitrogen to keep it that productive? Probably not. But if it's supporting that much production, and you are getting some oxygen, which means it's excessive, it's not all going into secondary production, because you are getting low oxygen, which I grant is true in that near field. But maybe it is still good for the fisheries, and I don't know how far you have to worry about cutting back to the point where that, starts to become an issue. But I would say that you could easily make some cuts and probably not affect that. I would still be in favor of cutting the nutrients back, maybe 20%. Don't ask me where I come up with that number. It's like 5,000. I mean, if they can say 5,000, I can say 20%. But I mean, I don't know. Wherever you would start, and then see if it starts having an effect. Sure.
Dissolved organic matter, in terms of its role in the hypoxic zone. Hypoxia is in the subthictal form. With the water in the subthictal layer, then does the dissolved organic matter not flock layers?
That's a really good question again, because I had people ask me that. They said, well, you're talking about dissolved organic matter. If it's coming in with the river is fresh water. How's that DOM going to get down to the bottom? I'm not saying the DOM is coming from the river. What I'm saying is that the DOM is coming from the marshes. Now, the marshes is where you can mix water, and we've watched water get mixed in the marshes, and you can get salinities up to 25, near 30 in some of the marshes. then that water is enriched with DOM, and when that water enters the shelf, you find it on the bottom. That's a good question, though, because if I was arguing that it was coming from the river, you've got this on the bottom, the river water is going to stay on the top. So no, it's not rivering DOM. But the marshes are shallow, it mixes, and when it goes out, it can sink and mix with it. So that's a good question. Yeah.
If it is that issue, and maybe they should just let your coast erode away because they were the marshes, and you won't have that DOM coming in there. The self-regulation.
Yeah, but don't tell the people of Louisiana that. They'll lynch you if you. They want to keep their soils, and it's bad enough that the Cajuns get flooded every now and then to save New Orleans. They're tired of hearing about saving New Orleans and being ignored and flooding their regions. But yeah, no, it's really that's the problem. I think the biggest problem. I mean, that's why I lost my home. Basically, you've lost all the barriers, when the storms come in, and that goes all the way out to the chandeliers where you have these large sand barriers from old lobe growth, and then the marshes. You've got all of those barriers that are now it's just open coast. So the storm surge comes right in, and so it's moving up further and further. I just think that's more of a problem than hypoxia. But if you've been measuring hypoxia for 20 years and you've been saying that same thing for 20 years, you can't change now, and they're stuck with that story, and that's why it hurts to now potentially have to change that if that's what you've been selling. And I think that's where we get into discussions where they just don't like hearing about it. So you have to kind of just sometimes admit maybe you were wrong. But I think I'm trying to stay neutral and stay out of it as much as I can right now. I've published my papers, and that's that.